Dear students, hope you all are doing well. This lecture is about another cellular organelle which is called as endoplasmic reticulum. As all the other cellular organelles, endoplasmic reticulum has its own importance, and it is a very important cellular organelle which is also involved in the synthesis of protein. Endoplasmic reticulum in biology or in the cell, it is a continuous membrane system that forms a series of the flattened sacs within the cytoplasm of the eukaryotic cells, and it performs or serves the multiple functions, being important particularly in the synthesis, folding, modification and the transport of the proteins. So endoplasmic reticulum is a membranous structure, and it is not made up of only one membrane. It is a continuous membranous structure which forms a series of sac-like appearance in the cytoplasm of the cell. All eukaryotic cells contain an endoplasmic reticulum. It is present in all higher organisms, in the cells of all higher organisms like all eukaryotic cells. In animal cells, the endoplasmic reticulum usually constitutes more than half of the membranous content of the cell. Differences in the certain physical and the functional characteristics distinguish the two types of the endoplasmic reticulum. So, on the basis of their physiological functions, their physical and metabolic functions, there are the two types of the endoplasmic reticulum are recognized. The one is known as rough endoplasmic reticulum. ER is abbreviated for endoplasmic reticulum, and the other one is smooth endoplasmic reticulum. Rough endoplasmic reticulum is due to its appearance. The rough endoplasmic reticulum has ribosomes on its surface. That is why it is called as a rough endoplasmic reticulum. And basically, the rough endoplasmic reticulum, because it has a ribosome on its surface, that is why it is involved in the synthesis of the protein. The other one is known as a smooth endoplasmic reticulum because it has a smooth surface and it has not present and it is not consisted of the ribosome on its surface. Like in the diagram, you can see this is the diagram of the endoplasmic reticulum. You can see it in this diagram. It is clearly found that the endoplasmic reticulum is a continuous membranous structure which is started from the nucleus and end to the plasma membrane. And you can see the ribosomes which are present on the surface of the rough endoplasmic reticulum, while on the other side, on the behind of the rough endoplasmic reticulum, there is a other type of the endoplasmic reticulum which do not contain the ribosome on its surface, and it is called as smooth endoplasmic reticulum. The membranes of the endoplasmic reticulum are called as the cisternae, as in the diagram it is mentioned over here, and the internal space of the cisternae is called as a cisternal space. And the main function of the rough endoplasmic reticulum is the synthesis, processing and the transport of the protein and the lipids. Smooth endoplasmic reticulum is involved in the formation of lipids. Here is a description about the rough endoplasmic reticulum. Rough endoplasmic reticulum is named for its rough appearance. I have told you before that its rough appearance is showing its the appearance of the rough endoplasmic reticulum is due to the presence of the ribosomes on its surface. So its rough name is due to its appearance, which is due to the ribosome attached to its outer surface. Rough endoplasmic reticulum lies immediately adjacent to the cell nucleus. It started immediately after the nucleus, and its membrane is continuous with the outer membrane of the nuclear envelope. So it started from the nuclear envelope and ends to the inner membrane of the plasma membrane or cell membrane. Ribosomes, which are present on the rough endoplasmic reticulum, they are specialized in the synthesis of the protein. that possess a signal sequence that directs them specifically to the endoplasmic reticulum for processing. So ribosomes, which are present on the rough endoplasmic reticulum, they make the protein when they get signal from the cell nucleus. When they get the signal from the cell nucleus in the form of the messenger RNA, then they start making protein and processing protein. A number of the other proteins in a cell, including those distant from the nucleus and the mitochondria, are targeted for the synthesis on the free ribosomes, or those not attached to the rough endoplasmic reticulum. Proteins synthesized by the rough endoplasmic reticulum have the specific final destinations. Some proteins, because every protein has to perform a specific function, so after their formation they have a specific destination. Some proteins remain attached within the endoplasmic reticulum, whereas the other are sent to the Golgi apparatus. Golgi apparatus are the cellular organelles which process or modify the proteins after their formation. So once the proteins are formed, they are in the rough endoplasmic reticulum, they are sent to the Golgi apparatus, which lies next to the endoplasmic reticulum. So after the formation of the proteins are sent to the Golgi apparatus, where further modification of the proteins are done. Proteins secreted from the Golgi apparatus are directed to the lysosomes or to the cell membrane. Lysosomes are the membrane-bounded cellular organelles which are responsible for which contain the digestive enzymes. So after Golgi apparatus, proteins are directed to the lysosomes, and still if there is any other modification required, so if all the modifications of the proteins are done, then they secrete to the outside of the cell. So after the formation of the proteins undergo various post-translational changes. Post-translational changes mean after their formation, proteins undergo various modification before arrive to their final destination. Proteins targeted for the transport to the Golgi apparatus are transferred from the ribosome on the rough endoplasmic reticulum into the rough endoplasmic reticulum lumen, which serve as the site of the protein folding, modification and the assembly. So after their formation in the rough endoplasmic reticulum, in which part of the Golgi apparatus proteins are modified? The lumen part of the Golgi apparatus proteins are modified for their proper functioning, because for their proper functioning, proteins after their formation, proteins need further modification. Modification may be of the various types. Suppose methylation, addition of the methyl group is required in the structure of the protein, or some sort of deletion may be required for the proper functioning of the protein. So everything is very much organized in our body by nature. Everything is organized for the proper functioning of our body, for the proper functioning of our cells. The proximity of the rough endoplasmic reticulum to the cell nucleus gives the endoplasmic reticulum unique control over the protein processing. The rough endoplasmic reticulum is able to rapidly send signals to the nucleus when the problems in the protein synthesis. Suppose in the synthesis of the protein there is a problem occur due to internal or the external environment. If there is any error occur in the synthesis of the protein, so there is a rapid signal generate which is sent to the cell nucleus, which is the control of the cell, and the rapid signal sent to the cell nucleus and the folding occur, and thereby influences the overall rate of the protein translation. When the misfolded or the unfolded proteins accumulate in the endoplasmic reticulum lumen, if in the cisternae space of the endoplasmic reticulum misfolded proteins are formed, a signaling mechanism known as the unfolded protein response is activated. If there is an error occur in the synthesis of the protein, so there is a certain there was a signal generate which is known as the unfolded protein response. It is also called as a UPR. That signal is activated in case of any error in the protein formation. The response is adaptive such that UPR activation triggers reduction in the protein synthesis. Because protein synthesis is not properly happening, so UPR signal activated. UPR signal unfolded protein response, which reduce or which terminate the synthesis of the protein and the enhancement in the endoplasmic reticulum protein folding capacity and endoplasmic reticulum associated protein degradation. Because the protein is not proper and if that improper or that protein which is not suitable, which has error, if that protein is formed, then the cell or our body would not function properly. So that protein is degraded by the cell if there is any error occur in the protein formation. So if endoplasmic reticulum if the adaptive response fails, cells are directed to undergo apoptosis. Suppose if the adaptive response is that which degrade the protein or which destroy that protein which has some sort of error. If that response is also not working, so in that case cells are directed to undergo apoptosis. Apoptosis is a phenomena in which a programmed cell death occurs. Cell kills itself if that error protein is formed. So the cell is going to kill itself just to protect the other cells, and that phenomena of the programmed cell death is called as the apoptosis. Now coming towards the other type of the endoplasmic reticulum, which is the smooth endoplasmic reticulum. By contrast of the rough endoplasmic reticulum, it is not associated with the ribosome and its function differ. The smooth endoplasmic reticulum is involved in the synthesis of the lipids, including the cholesterol and the phospholipids, which are used in the production of the new cellular membrane. You all know the lipid is a major portion of all cellular membranes. In the certain cell types, smooth endoplasmic reticulum plays an important role in the synthesis of the various types of the hormones like steroid hormones from cholesterol. In the cells of the livers, it contributes to the detoxification of the drugs and the harmful chemicals. The sarcoplasmic reticulum is a special type of the smooth endoplasmic reticulum that regulates the calcium ion concentration in the cytoplasm of the striated muscle cells. So there is a special type of the endoplasmic reticulum which is the sarcoplasmic reticulum, and it is associated with the regulation of the calcium ion concentration in the striated muscle cells. What are the striated muscle cells? Striated muscle cells are the cardiac cells and the skeletal cells. So it is all about endoplasmic reticulum, which has the two types, rough and the smooth endoplasmic reticulum, and both have their own function.